it is my pleasure to introduce to you two of my wonderful nature nerd pals and two of the wonderful people that I collaborate for the iNaturalist calls monthly. So wonderful wealth of knowledge. So let's get started. Deborah.
Hi, I am Deborah Barber. I work with the Nature Conservancy, and I've been using iNaturalist for, I guess, about 8 years now. Um, during the day, I direct land protection and land management for the nature preserves that we hold in the state of Maryland. A lot of ecological restoration, prescribed burns, tree plantings, invasives control. And iNaturalist really adds to my professional experience because it helps me keep records of what I'm seeing. I really enjoy working with Anna and Saranella on providing resources for and organizing the local community of people who use iNaturalist. I also use iNaturalist in my garden to record phenology, to learn about the weeds and the bugs that I'm seeing. And I really love using iNaturalist when I travel, when I'm in an area that is unknown to me and the organisms are unfamiliar to me. iNaturalist takes the place of a suitcase full of field guides. So it has added so much to my life. I am a big fan, and I'm happy to talk about it with you tonight.
And many of you know me already, but for those who don't, I am the facility director at Mount Rainier Nature Center, part of the Department of Parks and Recreation from Prince George's County during the day. That's what pays my bills. And then at night, I have the pleasure to be your program chair and night hike leader. I'm also born and raised in Puerto Rico, so if you want to talk mushrooms in Spanish, yo estoy dispuesta en cualquier momento.
Hey everybody, I'm Anna Kahanui. I have put my name phonetically on the slide in case you're curious on how to say my last name. I'm the co-founder of Capital Nature, a nonprofit here in DC. We promote the unique nature opportunities in the DC metro area. I'm a certified master naturalist in Fairfax County, and so I love being a nature nerd all around the DMV. My day job is at the U.S. Green Building Council. I've been there for 23 years. So if you're familiar with LEED buildings, I've been doing sustainability work for the last 23 years. I'm a certified forest therapy guide. So that means I lead forest bathing walks in the DC metro area for lots of different clients. So if anybody's interested in some forest bathing walks, let me know, or I'll put a link to my website that has the list of my walks if you'd love to see you out in nature. And with these ladies, as Deborah and Saranella mentioned, love being organizers for the City Nature Challenge here in DC. This is our 9th year. We always do so well. I think this year we have about maybe 690 cities worldwide, and I really hope that we always come in the top 10, which is great. Also, the Washington Area Citizen Science Network, a bunch of nature nerds that like to talk about citizen and community science. I'm originally from Honolulu, Hawaii, but I live in Fairfax with my dog and my husband, and that's me. So, what do community scientists do? Actually, can we go back one slide?
Sorry about that.
Yeah, no problem. So what is community science? Actually, it's also known as citizen science, and most recently, they're calling it participatory science. So it's been changing just to be more inclusive. And so, you have all these scientists with all these hypotheses out there in the world that want to gather information, but there aren't just enough eyes and ears and hands to collect all the data. And so, with curious people like ourselves, people like to be out in nature and make observations. Community science is when we all go out and gather the data to help advance this research. And so what's great about it is anybody can do it. You don't have to have a degree in anything. It's as long as you have, eyes, ears, a way to record some information, you can be a community scientist next. Next slide. So primarily, it's usually a volunteer gig. So a lot of us do a lot of volunteer service, and since a lot of us are master naturalists or master gardeners, you can log in some time doing your community science work. Basically, you're going to be observing things, listening to things, drawing things, taking photos of things, collecting specimens. So there's many different ways. Like here, I think we're looking at a mussel count here with Anacostia Watershed Society, where they're looking at tagged mussels. So there's many different ways that people can engage. Next slide. Now, why is it important? Well, we now know that everybody needs nature now more than ever. So this is just one of the many ways that people can connect to nature. I am a photographer, and when I learned about 10 years ago that my photos could contribute to scientific research, I got really excited about that, and I thought everyone should be doing it, which is why I love to spread the good word of iNaturalist, because I know so many people take pictures with their phones and devices, and if we could just gather all that and contribute to science, I just think everyone should be doing it. And then it's helping not only us as a community, but it's also helping the environment. Next slide. And really, it's about having fun too. So I have made some really amazing friends and connections being a community scientist. I love learning. Wonder is my favorite word. Wonder and joy are my two favorite words. So, being able to experience that, I mean, this is just one way to do it. So we just really want to, and we're basically preaching to the choir because you guys all like nature already, we know. But anyway, that's why we do it. I'm going to pass it over to Deborah.
All right, so a little bit more about what iNaturalist is. First of all, as my favorite citizen science platform, there are many, but I feel like iNaturalist does just about everything that I need it to do, and it does it really, really well. I use iNaturalist to record what I see in nature, both remote areas and in my neighborhood and in my garden. I've met wonderful other nature lovers like Anna and Saranella through my use of iNaturalist, because it's also a social platform. I learn more about the natural world. iNaturalist has made me a better observer and a better naturalist. And as Anna mentioned, my observations of the things that I see when I'm in the field, they don't just serve me, they contribute to science. They contribute to our global understanding of biodiversity. Next slide. So iNaturalist is super, super easy to use with a smartphone. But you also see a camera in this photo. If you are not a smartphone user and you are a camera user and you also have a computer at home, you can use iNaturalist that way as well. It's easier with a smartphone because smartphones automatically geotag and add time location to your photos, and those two pieces of information are important elements of an iNaturalist observation. Next slide. All right, so you will get started by making an iNaturalist account. It's a free app, and make yourself a username and you can get started right away. I was participating in a volunteer tree planting over the weekend. I mentioned iNaturalist, and standing right there in the field, this person was so excited about it that he downloaded the iNaturalist app and he started using it within 5 minutes. So that's how easy it is to get started. Next slide. All right, so I mentioned iNaturalist works on your smartphones, both iPhone and Android. It works on tablets, and it works on the desktop as well. Next slide. In fact, you've already seen a little bit of what iNaturalist looks like on the desktop. Earlier in this evening's lineup, we saw an iNaturalist screen from the desktop showing where people had found various kinds of morels in the Washington area. So iNaturalist does amazing things on the smartphone, but it does even more on the desktop. So a couple pro tips for you when you are using iNaturalist on a smartphone. I said you can look at the image on the left here and see different choices that you have. On the right, we highly recommend that you turn off automatic upload for a couple of reasons. That will save you on data. If you don't have an unlimited data plan and you are using automatic upload with iNaturalist, then a lot of information is going up to the internet while you're standing there in the field, and that can cause your data use to rack up. In addition, I find that iNaturalist uses more battery if I'm doing automatic upload. So my preferred way to use iNaturalist in the field is just take pictures and then upload them in the comfort of my own home in the evening when I'm not getting the sky causing a lot of glare on my smartphone screen, and my data on my home network is free. Next slide. I mentioned a couple of very important kinds of information that go into an observation. So iNaturalist captures who you are. It will show your username to other iNaturalist users. I'm comfortable having my username be similar to my own name. My iNaturalist name is D. Barber, but there are other people who have clever and fun and funny usernames that you would not be able to figure out somebody's actual legal name from their username. That is entirely your choice. iNaturalist also is a record of what you saw. I mentioned if you're using a smartphone, when you saw it and where you saw it is automatically uploaded with the photo or the audio segment that you're uploading. And the evidence of what you saw is very important if you want others to participate in identifying your observation. And the evidence could be a photo or it could be an audio clip. Next slide. So this is what an iNaturalist observation looks like on the desktop. This is an observation of eastern painted turtle by our friend Anna. And you see that there's a lot of information shown here in this observation. We have the photo itself, and then in fact, you can see underneath the large photo that she's got multiple photos. You see on the right side, you see Anna's username. You see how many observations she had at the time that she uploaded that observation. You see the location of this observation, which is the big blue pin, and you also see the other observations of that same species that have been made in the area. Looking at the bottom left now, we see that that Anna originally identified this as an eastern painted turtle, and then Luca Reptile came in and confirmed Anna's observation of that species, allowing that observation to reach research grade, as you can see from the little green banner at the very top of the observation where it says research grade. If you look at the bottom right, you see that reiterating the information that we have two agreeing. Oh, thanks for the pointer. You see that we have two agreeing species identifications for this observation, which means it's unanimous. Two people said it's eastern painted turtle, nobody disagreed, so that's what's leading it to be research grade. And when an observation is research grade, it can get sucked up, beamed up every so often to GBIF, the Global Biodiversity Information Facility, which is a global database of species all over the world. And researchers can download that GBIF information, and that information is used in hundreds of scholarly publications. So that's how your iNaturalist observations are contributing to science. Next slide. All right, so super easy to get started making an observation with a smartphone. You can take a picture either within the app, or if you're like me, you just take pictures and then when you go home, you upload them. So let's look at the second image here. You see that we've got a little thumbnail of the photo. You've got the field saying, what did you see? And then you can click in that field and iNaturalist will make suggestions about what that species is, what it thinks that species is based on visual similarity. Using some really smart computer vision technology, and that computer vision model is retrained monthly, so it's getting better and better and better. It's always been very good at identifying plants, and it is all the time getting better at identifying the less observed organisms like insects, spiders, and fungi. So let's take a look at the second screen again. You see all that other information that came up with the photo. You see the time and date. You see the location that photo was taken at. You see that the geoprivacy here is set at open. You have options there. You don't have to just take open, which shows the exact location. You can obscure it, or you can make the geolocation private. Anna has had the opportunity to mark this observation as captive or cultivated if she thinks it was planted, but she didn't do that. And she also has the opportunity to add it to a project. If we look at the third screen, you'll see what it looks like when iNaturalist is suggesting species identifications. You see that these species identifications, it says whether those suggestions were made based only on visual similarity or whether that species has been seen nearby or both. So those are all things that you can that you should take a look at and kind of study as you're uploading your observations. It is very important not to just use iNaturalist to get identification suggestions. That's just you being a consumer. Please be a producer. Please contribute to global science by hitting share or hitting the green check mark so that you're actually uploading your observation to iNaturalist. Because when you upload it, that's when the really, really magic stuff happens. When you upload it, your observation, your evidence goes in front of the eyes of iNaturalist users worldwide. And that's when the community of real people comes in and helps to refine your species identification. That's when it gets really exciting. Next slide. So all of these photos together, as I mentioned, are used to retrain on a monthly basis the computer vision model. So that's why the species suggestions that iNaturalist provides are getting better all the time. And I'm just astonished at how good they have gotten with decent quality photos. Next slide. So taking a closer look, if iNaturalist isn't feeling confident about the exact species, it will often at least suggest a genus or sometimes even a broader taxon like a family. But what you can do to do your due diligence when iNaturalist is suggesting species is click that little blue eye in a circle to the right of each species suggestion. That eye will get you to a page on each of those species that gives you more images that you can scroll through to compare with your image. It'll show you where your observation is in relation to, where your observation is in relation to the other reported locations of that species on iNaturalist. And you often can see a little thumbnail description of that species from Wikipedia. So it's really it's a really good step to take. If you're looking at all these different violets it's suggesting, you might not feel like just those pictures are quite enough for you to choose one over the other. So that little blue eye is your magic key to get you to more information. Next slide. All right, so a verifiable observation is one that has a date and is georeferenced, and your smartphone will take care of that automatically for you, has evidence in the form of one or more photos or sounds. You can make little audio recordings in iNaturalist and upload those. There is not automatic species suggestions for sound clips, and as far as I know, still no plans for iNaturalist to bring on that functionality. But that's where the community of iNaturalist citizen scientists come in, and they can help make suggestions. And then a research grade observation is not captive or cultivated. Research grade observations, well, if an organism is marked as captive or cultivated, it is not eligible for research grade because we don't want to confuse the database about all the things that are planted in gardens. Okay, next slide. All right, so here you see again the levels of identification. Needs ID, meaning there is not a consensus yet about what the species is. Casual is something that either doesn't have evidence or is captive or cultivated, so it cannot reach research grade, and there's research grade. Next slide. All right, I mentioned you can select your geoprivacy. The default is open, but you can obscure the geoprivacy. Maybe when you found a choice morel patch, you don't want to share the exact location with other people on iNaturalist. That is your choice. Another reason to obscure geoprivacy is you're on private land that is not your own. You're visiting a friend and you don't want this showing up as in your friend's woods. I want you to know also that iNaturalist automatically obscures the geoprivacy on sensitive species, such as rare, threatened, and endangered species, and also some species that are not rare yet, but that are charismatic and potentially vulnerable to collection, like eastern box turtles. You can also set the privacy on geoprivacy to completely private, in which case nobody will even know what continent your observation is on. So it will be very difficult to get the community of identifiers to agree with or refine your identifications if they don't even know that your observation was made in Arlington, Virginia, or Antarctica. So I do not use private geoprivacy at all, but I often use obscured geoprivacy. Next slide. All right, so all of these data points that are shared openly contribute to research and conservation around the world. Excuse me. And that information has added a lot to science. Just as a small example, on the next slide, you can see how all this information, next slide, you can see how all this information in aggregate can be used to show that some things are flowering earlier. Perhaps, we suspect, in response to warmer springs that are happening because of climate change. Next slide. Excuse me, getting a little hoarse. Okay, so there are lots of ways that you can get help. When you get onto iNaturalist on the desktop, you can easily get a link to the iNaturalist forum where you can post your advanced questions. Within any iNaturalist observation, you can tag any iNaturalist user by mentioning them in the comments section. So you can tag Anna, Serenella, me, anybody you know who's using iNaturalist if you want that observation to get their attention. And here in the DC metro area, we have a monthly event which used to be the first Tuesday, but now it's the second Tuesday, so Serenella does not have to miss these meetings with her fungi friends. And we get together on every second Tuesday. Anna, Serenella, and I usually host a presentation from an outside speaker and then spend whatever time we have available working together on identifications on iNaturalist. So it's a lot of fun. You meet a lot of cool people, and it's a great sense of camaraderie. So you can see more about those at the link shown at the bottom of the slide. Next. All right, I'm going to send it to Anna now.
Thanks so much, Deborah. Okay, so what should we observe? Well, I'm gonna wonder what this group might want to observe during the City Nature Challenge, but I'll give you a couple other examples in case you're interested. I personally, when I'm talking to people promoting the use of iNaturalist, I like to tell people to start with things that they love. If there are things that they're already observing and already taking notice of and taking pictures of, for example, I am a fungi fanatic and I did join the club this year, so I'm excited to be a new member. I love butterflies, pollinators, bees, flowers. So I just encourage, if you're new to this, just start, don't feel like you have to go crazy and document all the biodiversity in your neighborhood or anything. Just start with something you love. Next slide. Deborah mentioned when we were looking at an example observation, the difference between wild and captive cultivated. Here on the left is my dog Lonnie. She is not a wild creature. She is a domesticated animal. So if I were to take a picture of her and put it on iNaturalist, I would definitely mark it captive cultivated. Now I probably wouldn't do that. I wouldn't waste everybody's time, but she's cute, so we put her in there. If you're at a botanic garden or at the bonsai garden, anywhere where there's tags of things where you know that things were planted, definitely you would want to mark them captive cultivated. Next slide. And then what's really great is they look for things on other things. So for example, I work with I live in Burke, Virginia. We've set up an iNaturalist project for their pollinator garden and their library property. And even though they planted their pollinator garden plants, those are all considered captive cultivated, but anything that's on and that visits those plants is fair game for this challenge because we're looking for wild species. And so that could be bugs, that could be this little spider. I think it's a crab spider. inside a teeny tiny pawpaw flower, which I didn't see the spider until hours later after I took the picture when I was enlarging it. So that was such a joy and a delight. Just finding little bugs and critters on leaves, possibly on trees that were planted, like say in your front yard, and then even things on the right, like we have a tar fungus there on a red maple. I know Saranella and I love it when we find like tar funguses on things. So yeah, so just everything on captive cultivated things could be fair game if they are wild. Next slide. I'm passing it to Saranella.
All right. So what are we going to be observing? Well, mushrooms, of course. And not all the pictures are created equal. Not all the pictures have the characters that allow you for a proper identification. So to give you the best chance at getting the information that we'll need to help you ID, here are some of the tips. And the first one is please, 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 please clean your camera lens because most often than not, what we're looking for is a very sharp picture. But if your camera lens is dirty, it's gonna obscure, it's gonna blur and obscure some of those characters that we are trying to see. And in here, we have a really nice example from Annie Weisman, Annie Dobbs, that showcases how she has set up the specimen to be observed, cleaning any debris that might be on top of the mushroom, and really creating a nice plate for us to observe the characters. And it's really important to be able to describe the habitat in which that mushroom is growing. And we'll see more about that in the next slide. And especially for bolids, excessive manipulation of the specimen should be avoided because sometimes they bruise. And if you manipulate it too much, then the untouched colors might be obscured, and what you end up is with the reaction only. So we want to see as much characters of the uh, original mushroom plus the reactions. So what I'm saying is be prepared to take multiple pictures. And we've already talked about making your observation. One reminder that I have for you is to make sure that you have activated the location services for your camera in your settings. That way, when you take your pictures, all the metadata will come up with your picture. That's the date, the time, and the location where that picture was taken. And when you are preparing to go out to the forest, make sure you are prepared for what you're about to encounter. So bring a bottle of water, have the GPS turned on your camera, and if you are working with a camera and not your phone, then you're gonna need definitely something to write so you can take notes of where you observe that particular mushroom. So when you go back home, you can approximate the location where you found that specimen to the best of your ability. So here is that uploading of the pictures again. Um, we were talking about the alternative of doing an iNaturalist observation in the forest versus an iNaturalist observation when you're going back home or when you're sitting in the car after the walk, which is what I do. Um, what I prefer doing is taking the series of observations on site and not using iNaturalist until I am back at my car or sitting back home. That allows me to maximize the use of the time that I'm in the forest to make the actual observation. And if your signal is weak, that doesn't impede the activity because you're just taking pictures with your camera. So you don't need that strong signal to be able to record everything that you need to see. What you do need is that sequence that you're going to take your pictures in. I like to start from 2 feet away and then get closer and then end my picture series with the voucher just so I have all the information in the same way. And when I am uploading the batches or of pictures, I know that right when the voucher hits, it's the end of my series, and the next picture is the start of the next one. Now, I have very good news for you. If you are an iNaturalist user, as I am, you will love to know that there is a new iNaturalist app out for beta testing, and it's called iNat Next. And with that iNaturalist app on your phone, unfortunately, is for iPhone users only, if I remember correctly. Um, this one allows you to do batch uploads of multiple observations from your phone. That is a quality that was only possible on the desktop version of iNaturalist, but is now able to be done on your phone with iNaturalist Next. I've been using it, and so far I love it. So I don't have to go and upload just one observation by itself. The caveat here is that so far you can only do batches of 20 pictures, which for me translates into 4 observations. However, it's an improvement, right? So I am really excited about the changes that are coming with the app. And here you can see an example of one of my series, what Deborah was mentioning. When you look at an observation, the bottom is going to show you the series of pictures that you've uploaded. And as you see here, I only saw one single cup. This was back in Puerto Rico. So I take multiple pictures. It's attached to wood. It's a little closer so I can see the margin and the texture and the underside and the top side. And then I finish the series with the voucher. This is sample number, whatever, the time, the date that I took this sample. And then this little envelope goes straight into the dehydrator so that all the information is with the specimen. So, friendly reminder that we have our champion observer, who is Annie Dobbs, and this is some of the information that you're going to see. For the fungal specimens that have been considered for DNA extractions, you are also going to see an annotations section where when the sequence comes back, we will put the sequence there for the ITS or barcoding gene. So there's a lot of good information that you'll be able to observe or learn from in the observation. And sometimes the mushrooms can be a little challenging, so it's really educational to read all the comments under the activity because different experts can come there and debate what characteristics of that particular mushroom lean the identification to one clade versus the other. That can be very, very educational, at least for me. So I really enjoy that part of iNaturalist. And the educator that I am, I have produced a list of things, a list of pictures for you to take when you're doing your mushroom identification in the forest in the order that they should be done, please, and thank you. So, before you even think about detaching your mushroom, please make sure that you take a picture that shows where is it growing from? What is the substrate? Is it attached to something alive? Is it growing from the ground? Is it growing on a dead log? Make sure that your picture captures that. If it is attached to something alive, identifying that something that the mushroom is attached to the best of your ability, it is very useful. And it doesn't even have to be down to the species level. If you can say, hey, this was attached to a holly tree, or this was attached to a conifer tree, as much information as you have to give, we would love to have it either in the comment section or shown in a picture. And then not only capture what is it growing on, but also how is it growing on that. Is it growing like a mass of mushrooms that are uh, joined together? Is it growing like a bouquet coming from a single point of origin and then branching out? Or is it we are a group of mushrooms that we're together but not really stuck together? All of those characters are very important to capture in your pictures. Once you've taken that, then take the classic picture from the top, please and thank you. Make sure that that picture from the top also has the details of that top, not just a blurry center or a blurry edges, but as much in focus as you're able to give. Once those are done, then you are ready to detach the mushroom and see the underside. We know that the spore producing surface of a mushroom can look vastly different from one species to the next. It can have pores, teeth, or gills, and it's only when you look under that you can see that. It's also important to capture if there is space in between the gills and the stem. So capturing the underside with clarity is very helpful. Cut through the spore producing surface to see if it's bleeding. Is it changing color? And then it's also good to get the base of that mushroom where it is attaching and sometimes digging a little bit and bringing it up to ensure that you capture the whole base. If you observe any smell or bruising that was not able to be captured in the pictures, please write that in the note section on your iNaturalist app. Now, I belong to the DNA committee, and I am super, super grateful and excited to see the enthusiasm that has been generating as of late of your efforts and bringing samples. And I have to do two big shout outs here to champion collectors. So the first one is Annie Dubs. She has brought so many wonderfully preserved samples for the research. And the second is Jennifer Pfeiffer. Hi, Jennifer. Thank you. So, um, these ladies really know how to make a great observation and a great sample. And I have some of the examples here. At this point, I don't care what you use to collect your sample as long as it is paper, so it allows airflow and has the right information annotated. So here are 4 samples of things that we have right now here in the DNA lab slash my office. And if you look at all these 4, you're going to see that the most important thing that you can add is the iNaturalist observation number. If there is not an iNaturalist observation number on the sample, I never saw it, it does not exist, and it will be added to the compost, please and thank you. Now, if you want your effort to count, please give us that iNaturalist observation because that is the unique identifier for that particular specimen. And that goes through the entire process. That is the number that we refer back to. From the minute we dry it to the minute that we get the sequence back is the iNaturalist number that keeps us in check through the whole thing, including your pictures that show us how beautiful that piece of dried, unrecognizable mushroom was. So it's really, really important to get the iNat observation number. Bonus points if you can give us a field ID. And as I was mentioning before, we don't expect you to be able to ID 2 species. God knows I cannot ID 2 species, a lot of things. So give us your best shot. If you think it's a Russula, great. If you think it's an agaricales, meaning it looks like a mushroom, fine as well. Just tell us what is your guess or the guess that iNaturalist gives you. It just helps in the sorting process of the samples. Um, and it's really good to have your iNaturalist handle on the samples because we know then if there is a dispute on the number, a mistranscription, then we know who can we go and ask. And the date is very helpful as well. So you see that both my sample and Dave Webb's sample here have the date, and that is helpful so we can cross-check all the data that we have. And I want to highlight how beautifully careful Dave Webb was in bringing his sample to us. He dried the moth infected with the entomopathogenic fungus and then put it in a vial so that we could have the best specimen as possible, which we appreciate. We don't expect you to go and buy anything. You can come to the Mount Rainier Nature Center to get your sample bags, your little boxes, or you can reuse some of those recyclable materials at home. As long as you have the information correctly cataloged in that sampling container, I'll be happy. And of course, we love well-documented samples, so thank you in advance. So Here is an example of what well-documented sample looks like. In the middle of the forest, I started with my series. Um, the collaging here doesn't have the order because of aesthetics, but you get the idea. So a picture from afar showing the attachment or the substrate from the top, from the side, a little closer to capture the details and the hairs on the stem. And then I ended with a picture of the actual mushroom with the voucher. So when I'm coming back home, I can look through the pictures and I know, okay, this series belongs to this particular mushroom. When I'm trying to go through the little actual samples, I have the pictures that say, this is what I'm supposed to have in this. So it just helps the scientific process. So what I'm trying to say is, please make sure that you get close when you're taking your observations to capture those very fine details. In the kingdom fungi and allies, macro photography is there's no substitute. Macro photography is king, even with micro photography needed in order to identify many of the individuals to the species level. So get comfortable, get close. Make sure your pictures are in focus. Clean your camera when you're with your cell phone. Touch the screen in the area that has the most surface for that particular specimen so you get the most focus out of your picture. And like I said, capture detail. If I didn't get close, I would have never seen that this tiny mushroom that is 1 inch tall has hairs at the top. Like, who's gonna see that? You can even use your magnifying lens over your phone camera lens in order to see this type of detail in your mushrooms. And it is not a foray in which I am if I'm not on the floor. I am queen of the little, and I happen to have my traffic director here with me tonight. I have laid in the middle of a trail to take pictures of a lichen and had Anna directing traffic over my body. I'm okay. I'm just taking pictures of mushrooms. So bringing a friend with you is very helpful. Ay, thank you, Anna, for being my traffic lead.
Anytime.
And a friendly reminder that we have here a focus in our research objectives, which are the rustula's and bolides. And for that, we have a picture from an observation of Matthew Bessiat. I hope I didn't murder that. And again, a picture of the top. Where is it growing? It's growing in the ground in the middle of leaf litter. And the underside, so we can see those scales. We can see that very stalky stem. And again, Martin Livesi shows us how a bolide observation is done, this time showing that color change when you bruise the spore surface of the bolete, changing from yellow to navy blue. And with boletes and many other mushrooms, it is useful when you cut across it so you can see how the stem on the inside looks like. Mitch was talking about that when he talked about morels versus Geomitra. So making that cut is very useful. And don't feel bad about making that cut, especially on as I left for a second. Everybody cool? Still here? Okay. The computer was trying to play tricks on me, and I went, no. Anyway,
We're still here.
um,
We only have about 5 minutes left though, so
okay. Anyway, so here is the showing the color change and on the mushroom and the spore producing surface. So really important to do that. And when you are looking for mushrooms, chances are you're gonna see the wonderful blooms that are right all over where they are. So if you are so inclined to take pictures of that for the City Nature Challenge, um, please make sure that you are capturing the leaf arrangement. And here at the bottom, I have what does that mean? It's just how the leaves are arranged around the stem. Um, Pro tip, the underside of the leaf can also be very informative. For arthropods, pro tip, take pictures that include the head, but also the abdomen. And we naturalists like to hang around with a Ziploc bag so you can capture that insect and turn it upside down and capture more information when you're taking pictures of insects. Um, Multiple angles, and sometimes when we're talking about crane flies and damsel flies, good luck with the damsel flies. Taking pictures of the venation of the wings are very informative. At the end of the presentation, I will put this link on the chat so you can get it because I know I am flying by for time. And don't be afraid to use your hand for scale if you don't have a tape measure. You don't have to pick up the spider, just putting your hand next to the spider is fine. Mitch, I saw your face. Anyway, so a reminder, please make sure that you are replacing any disturbed microhabitats. Bring a phone charger if you plan to stay more than an hour or two outside so you are still connected and you have what you need. And bringing a small ruler, a coin, um, or your hand for scale into the picture, it's always a good idea. And Anna, if you can give a very brief
Yeah,
version of this.
I'm just gonna do a speed round really quickly. So I'm sure most people on this call know what a BioBlitz is. That's what the City Nature Challenge is. Senses of nature, 4 days period of time. We're all trying to get as much stuff in wild observations as possible. Next slide. So it's coming up. These are the websites. The one on the left is the global website. If you just add DC in the end, that is our DC website, which is lovingly curated by our friend Deborah Barber. Next slide. And the folks at the global office put together this kind of cute little slide. So if you're trying to tell your friends and buddies about it, this is just basically the gist of how it works. I won't go through all the different numbers, but it's basically do the app, take the pictures, find stuff, and upload it. Next slide. These are the dates. So it always goes from a Friday to a Monday. I think the city count is closer to 690 now. And so we'll be doing it everywhere from people's backyards to community parks, gardens, anywhere where there's wild nature. Next slide. And I don't even need to explain why, it's because it's cool, because it's fun, because you'll meet people, because you'll learn stuff, and you're going to help highlight our DC metro area's biodiversity. Next slide. Just so you know, our count happens to be the DC metro area boundaries. So if you're in any of these counties in Virginia, Maryland, the one county in West Virginia, anything that you make observationally that happens within those bounds will count for our challenge. You don't even have to join the project. Next slide. These are the important dates. These are some of the many organizations that participate with so many different, great ways to network and meet with other organizations. Next slide. Just take a quick glance. I think we'll be sharing these with you, but check out from 2 cities starting 10 years ago up until 690 cities this year. It's a just astronomical growth in terms of what's been going on with the City Nature Challenge. Next slide. These were the observations made totals for last year, 2.4 million, 65,000 species, 83,000 people. Next slide. And then DC, as I mentioned earlier, we are always in the top 10. So we were 8th for observations. Next slide. We were 11th for species. Oh, actually, maybe we didn't make top 10. Okay, so I stand corrected. 11th for species last year. Next slide.
Our fungi friends are gonna help us go back to 10.
Yeah.
Yeah.
6th for people. We always are great in terms of people engagement and community science. Next slide. These are the top 10 species. Let's get some fungi up there, people. That would be awesome. And then just the highlights and stuff. Tag us on social media and get out there and practice. And then I think the next slide is about swag. Yeah. So if you want a T-shirt, a cool T-shirt, go please order one and wear one with pride. I think that's it for me. And then I think we can just move on to the Deborah can mention the events.
Okay,
Can
time check.
you-
We got 2 minutes, just
Can you
FYI.
can you go back one slide and let Deborah talk about the map real quick?
Yeah, so if you go to citynaturechallengedc.org, there's a field events page that lists all the details you need to join any of these events, and people are adding more events. Actually, this is last year's map, but this year's map is equally fabulous. Lots of good field events to join. And if you could just go through the next few slides real quick, we'll make any highlight. Yeah, we've got some wonderful nighttime event at the very end of the City Nature Challenge. Next, Kenilworth Aquatic Gardens. So many organizations participating. I'm so proud of the way our community is putting on wonderful events for people to attend. Okay, you can follow either of these QR codes to get to these websites that have a lot more information. And then after the observation period, we really work on identifications. And to that end, we're organizing a virtual ID party. You can join us after acquainting you with some of the really great tools that exist on the desktop to help you compare one species with another. We go into virtual breakout rooms and focus on the taxon of our choice. So it's a lot of fun to work in collaboration and partnership with other members of the identifier community on iNaturalist. All right, I think that is it.
Yep, I think that's it.
Awesome. Thank
Thank
you,
you so
guys.
much, Deborah and Anna. Floor is yours, April.
Yeah, this was fantastic as always. I'm really excited to see a good turnout this year. And look forward to everybody participating. And I'm very excited to see what we find out there. And with that, we're going to close out, and we will see you either next month or sometime with the events coming up, including the one tomorrow night. Hope to see some of you for our happy hour, and thanks again for joining tonight. Take care, everyone. Happy spring, happy morel hunting,
Happy iNaturalist.
and
Thank you.
happy iNatting. I love
If
that
you
verb.
have questions and want to stay behind, we will stay to answer those.